It's my pleasure to introduce Dr. Chris Breuer, who is a professor of surgery at Nationwide Children's Hospital. He's also a nationwide endowed chair in surgical research, and he is also a board-certified pediatric surgeon, so a physician scientist, runs a lab as well, as you'll see, does some really exciting work on vascular tissue engineering, has a number of awards, but what is really exciting, I think the most exciting, and why we invited him here today and really excited to hear his talk, is him and his close collaborator were the very first people to tissue engineer a blood vessel that was then implanted into a human infant. And has been working on that goal to develop vascular grafts that will grow with kids, which is a big issue. So I think really exciting example for translation, and I'm glad you could join us today.
Thank you very much, Karen, and thank you very much to the organizers for the introduction to participate in today's meeting. As was mentioned, my name is Chris Breuer. I'm a pediatric surgeon at Nationwide Children's Hospital and a physician scientist. And what I'd like to talk to you today is about the development and translation of a tissue-engineered vascular graft for use in children. And I'd like to disclose that some of the work presented today was funded by some of my industrial partners. So in general, if you're a child and you need a vascular graft, it typically means that you have a congenital cardiac anomaly because congenital heart surgery is the most common cause or need for vascular surgery in children. And congenital cardiac anomalies affect about 1% of the population, and about a third of these are critical, meaning that they're life-threatening without urgent surgical intervention during the newborn period. And when a patient has a critical cardiac anomaly, the surgeon needs to reconstruct the heart. And due to the paucity of tissue resulting from the congenital cardiac anomaly, it typically requires the use of biomaterials, either in the form of vascular grafts, vascular patches, or replacement heart valves. And no matter what the biomaterial, they're all associated with problems, whether they're synthetic or biologic, as imaged here. These materials are all susceptible to infection. They have a great propensity to calcify over time. They have poor durability due to problems like neointimal hyperplasia. But perhaps most fundamentally, they lack growth capacity. So a child can have a life-saving operation only to outgrow that operation and require additional surgery and incur additional risks. Now, occasionally, or probably better rarely, the surgeon can perform this operation using purely autologous tissue, so without the need for biomaterials. And when the surgeon could do this, they can avoid all these complications and potentially improve outcomes. So the premise underlying our work is that we can use tissue engineering methods to create autologous tissues with the hope that they'll behave more like our own tissues and less like the synthetic materials that we're forced to implant. And tissue engineering can be defined in a variety of ways. The way I look at tissue engineering is it's a science that attempts to leverage the fact that our own cells have the ability to divide, multiply, and then self-organize into functional units. And one method of tissue engineering, the one we use, uses a biodegradable scaffold that can serve as a template to direct the tissue formation. And then the resulting neotissues, which are autologous or made from the patient's own cells, can be implanted and used in the surgical field. Now, I've been at this for a long time, probably a little bit too long. I actually started the project I'm talking to you about today in 1995 when I met my collaborator, Dr. Toshi Shinoka, and we've worked on this project ever since then. And at the time, we were introduced to what's now called the classic tissue engineering paradigm. And literally, when you came into the lab, you sort of selected an organ. And what you would do is you'd harvest a piece of that organ, you'd isolate the cells, you'd try and expand those cells in culture, you'd seed them onto one of our scaffolds. You might do something on that seeded scaffold in vitro, you might just implant it, and then you'd see if you could create the tissue. And so we used the Model T of all scaffolds, the polyglycolic acid felt sheet, and we tried to make blood vessels from it. And at the end of my time in the laboratory, we were making nice blood vessels that could be implanted in animals, and they were functional, so we were quite excited. But all good things must come to an end, so completed the postdoctoral fellowship, and Toshi and I each went our own ways. Toshi returned to Tokyo Women's Hospital, his home institution, and he started his own lab in addition to his own practice as a congenital heart surgeon, and started to work on developing and making these grafts. At the time, he also started to collaborate with the engineers at the Gunzai Corporation. The Gunzai Corporation is to Japan what Fruit of the Loom is to the United States. It's the big underwear producer, but they also happen to be the largest manufacturer of biodegradable medical goods in Asia. And along with the engineers at Gunzai, they refined the design of the scaffold. So they created a knitted polyglycolic acid tube that was then coated with a 50-50 copolymer of polycaprolactone and polylactic acid. This was designed to degrade by hydrolysis over about a 6-month period, and it had excellent surgical handling capabilities. And Toshi started to use the scaffold in his animal work, and when he got to the point that he felt comfortable that it could create blood vessels, he went to his institutional review board and got permission to implant one of these in one of his patients. So this was before the time where regulatory agencies were even involved with this process. And in 2001, he published the results of the first implantation of a tissue-engineered vascular graft, and it was used to repair the pulmonary artery in a little baby undergoing congenital heart surgery. The patient did quite well and continues to do well to this day. As he tried to continue with this work, though, he noted problems. He was having difficulty harvesting tissues and creating enough cells to make constructs. So frequently patients would have a saphenous vein biopsy, but not have adequate cells to then implant the grafts that he was trying to construct. So he returned to the laboratory and started to investigate other potential cell sources. He had read a paper that if you seeded polytetrafluoroethylene grafts with bone marrow, that you could increase the ingrowth of endothelial cells. So based on this finding, he thought maybe the bone marrow cells could serve as a source of seeding for creating tissue-engineered vascular grafts. And he began to look at the different fractions of the cells from the bone marrow and discovered that the mononuclear fraction of cells could be seeded onto the scaffold and obtain the same results in his animal work that he obtained by seeding endothelial cells and smooth muscle cells onto that scaffold, the cells that are typically used to make a blood vessel. And he thought that this might be due to stem cells within the bone marrow. But again, under IRB protocol, again, there's no regulatory agencies working on this at this time. He starts the first clinical trial looking at the use of tissue-engineered vascular grafts in an operation called the Fontan operation. And this is an operation that's used to treat children that are born with what are called single ventricle cardiac anomalies, probably the most severe form of congenital heart defect. And in this operation, you need a large vascular graft to connect the inferior vena cava to the pulmonary artery. And he enrolled 25 patients into the study, and the study basically generated the feasibility of using tissue engineering techniques in humans. He demonstrated through serial imaging that the grafts tended to increase in size over time, suggesting that they had growth potential, and he had no graft-related deaths. He did discover that the most common graft-related complication was graft narrowing, which is called stenosis. Now, while Toshi was doing the translational work, I started my career at Yale University, and Yale had created a tissue engineering center with a slightly different focus. They were more interested in understanding how things worked on a cellular and molecular level. So I started my laboratory and wanted to understand how a biodegradable scaffold seeded with bone marrow cells could transform into what we call a neovessel, the tissue-engineered vascular graft after the scaffolding's degraded, which has an intima, a media, and an adventitia, and really looked and behaved like a real blood vessel. And to this end, I decided to try and create methods for implanting the grafts in mice so we could take advantage of transgenics and start to do things like cell tracking experiments, which had never previously been done. So we developed methods for fabricating our scaffolds on a much smaller scale. The largest blood vessel in a mouse is a little bit less than a millimeter, and then developed microsurgical techniques for implanting these scaffolds. And what we discovered is that the tissue-engineered vascular grafts could form neovessels in the mice just like they were doing in our large animal studies. And that over the course of about a 6-month period, the period of time it takes for the scaffold to degrade, we could create these neovessels. And over the course of about 2 decades, I systematically dissected what was going on and had what I would characterize as probably 3 important discoveries. The first important discovery is that we did the right thing for the wrong reason. As I mentioned, when Toshi started to seed the bone marrow cells onto the scaffold, he had assumed that they were a rich source of stem cells. But when we actually did the cell tracking experiments where we put labeled bone marrow cells onto our scaffold, we discovered that not only did they disappear, but they disappeared really rapidly. They were gone within 24 hours after implantation. The second thing we discovered is that the endothelial cells and smooth muscle cells, the blood vessel cells, actually came from the neighboring blood vessels. So the cells were migrating along the luminal surface of the scaffold and creating the vascular neotissue. And then finally, we discovered that this whole process was inflammation driven, that if you implanted these tissue engineered vascular grafts into knockout mice where you could knock out different inflammatory cell types like the monocytes and macrophages, you could actually stop the whole process of vascular neotissue formation. And armed with that, it enabled us to more systematically and rationally sort of move forward with the design and development of our product. So we really began to focus looking in on the problem of stenosis. So as I mentioned in Toshi's original clinical trial, we discovered that stenosis was the most common graft-related complication, affecting about a third of the patients over the lifetime of the graft implantation. And none of the grafts failed because of the stenosis, but it was something that we wanted to improve upon. And to simplify things, what we discovered is that stenosis was due to excessive inflammation, and that the cell seeding actually functioned as an immunomodulator. By seeding those bone marrow cells onto the scaffold through a paracrine signaling mechanism, they could reduce the amount of monocyte and macrophage infiltration to the scaffold, which also reduced the incidence of stenosis. So in 2008, I had an opportunity to recruit Toshi back to the United States, and we started our own laboratory. And one of the first things we did is we went to the FDA, and we said, we have a technology that we're excited about. We think it has the potential to help patients, and we want to work with you to see if we can bring this to the clinic. And the FDA was quite excited about the indication. They liked the fact that we were using this in the Fontan operation because this was a relatively safe initial clinical indication. Now, the Fontan operation requires a large caliber graft in a high flow situation. Large caliber, high flow equals low chance of clotting. So they thought that that was great. And then the other advantage of the Fontan circulation, it's a low pressure circulation. It connects a vein to the pulmonary artery, so low pressure, so less chance of aneurysmal dilatation and graft rupture. So the combination of those two things they thought was really good. And then the last part they liked about it was that there were no products available that could grow. So we could meet a unique need. And when they reviewed our data, they really liked the scaffold. The scaffold was manufactured using GMP techniques. They were happy with the way that we were manufacturing. They were satisfied with that. But they were horrified by the way we actually assembled the grafts. So in Japan, when they made one of these grafts, they'd harvest the bone marrow, they'd isolate the mononuclear cells, and then they'd pour the mononuclear cells onto the hydrophobic scaffold, and then you'd massage the scaffold with your fingers because that would help the cells go inside. They didn't like this. So they gave us a mission. They said, look, if you want to try and do this in the United States, you got to come up with an operator-independent method for seeding the scaffolds, and you have to do everything under GMP conditions, not only manufacture your scaffold. So 3 years and 3,000 pages later, quite literally, we had approval to do the first implantation of a tissue-engineered vascular graft. And so in 2011, we implanted the first tissue-engineered vascular graft in the United States, and it was in this little girl. Her name's Angela, and Angela's about 6 months after surgery, and fortunately her operation went well. Now, at this point, we opted to leave Yale University and go to Nationwide Children's Hospital, which is in Ohio. And this was driven by really two factors. We needed to be in a bigger congenital heart program so we could have more patients to enroll in our study. And Nationwide had particularly good clean rooms and regulatory expertise that was available to help us move this technology forward. So we got everything set up and continued our trial. And unfortunately, the next 3 patients all had very, very similar complications. They all developed graft narrowing or graft stenosis, which you see on this angiogram. And all patients at about 6 months after surgery that developed this significant stenosis were taken to the cath lab and underwent balloon angioplasty. And fortunately, the grafts could be salvaged. But because of this high incidence of stenosis, higher than we had predicted would happen, the FDA put our clinical trial on hold, and ultimately we closed the trial prematurely. But we continued to follow the patients, and the patients continued to do really well. In fact, all of these kids now are between 10 and 13 years after surgery and have had no additional graft-related complications. And more importantly, serial imaging with MRI demonstrated that as they've gotten bigger, their grafts have grown with them. And as we started to analyze the data and look at the results, we noticed that there were really two phases of tissue formation. There's this first 6-month phase where you had a scaffold, a biodegradable scaffold in place, and the tissue's forming on the scaffold as that scaffold's degrading. And then there's the neovessel phase, where after the scaffolding's degraded and we have this biologic structure, they behave completely differently. During the neovessel phase, or phase 1, the grafts are dilating, they're narrowing, things are happening very rapidly. During phase 2, after that scaffolding's gone, it's more, much steadier, much more gradual changes. And so I started to wonder about how this process occurred and if there were better ways of understanding it. And around this time, I kicked off a collaboration with Jay Humphreys. Jay Humphreys is a biomedical engineer at Yale University who does quite a bit of computational modeling. And Jay revealed to me that when he looks at the world, he sort of sees equations, which is funny because that's not what I see. And Jay had spent the previous 20 years developing computational models that could quite nicely describe and predict how a native blood vessel would behave. And his modeling was all predicated on this thing called mechanical homeostasis. And what mechanical homeostasis refers to is the fact that our own smooth muscle cells sense the hemodynamic forces that are being applied to them. And there's a certain amount of mechanical force that those cells need to sense in order to be quiescent. And if that's perturbed, if they're either over or understimulated by the hemodynamic forces, those cells will react. And they react in a very predictable manner. In the short term, they react by either vasodilating or vasoconstricting, and that vasodilation or vasoconstriction changes the flow of blood so that they sense that same, or they change the flow of blood such that the mechanical forces that they're sensing are the amount of mechanical forces they need to be quiescent. In the long term, what they do is they either break down or remodel the extracellular matrix to either stress shield or not stress shield the tissue. So if you can measure the hemodynamics and you can determine the set point, that amount of mechanical force that those cells want to sense, you can actually quite accurately predict what will happen. And what Jay thought is that in order to do this for our tissue-engineered vascular grafts, we need to add an extra term to his formula, to his computational model that would describe this inflammation-driven tissue formation that I had described to you, that the presence of the scaffolding material was what was driving the inflammation. So if we could combine these two things, we could create a computational model, which we were able to do. And as we began to run simulations with the model, what we discovered is that the model very accurately predicted the stenosis that we experienced in our clinical trial. But what the model also predicted was that stenosis would get better by itself without any sort of intervention like angioplasty. And this was strange because this doesn't normally happen in the clinic. If you put in a Gore-Tex graft and it gets stenotic, you either have to angioplast it or remove it. So it was something that we'd never considered. So it was exciting because it gave us a chance to validate the model experimentally. So we did a large animal study where we implanted a large number of these tissue-engineered vascular grafts into lambs and then serially monitored the development of neovessel formation using serial angiography. And here we see an image of the graft 1 week after implantation. By 6 weeks after implantation, 25 out of 25 of these lambs all developed tight stenosis, just like we'd seen in our clinical trial. But we didn't do anything about it because the model said it would go away, and lo and behold, the model was right. It went away without any sort of intervention, and by a year after implantation, the resulting neovessels were behaving like regular neovessels. Furthermore, the lambs really tolerated this fairly well with minimal sort of complications. So what we discovered, in essence, is that this narrowing was a part of the natural history of neotissue formation. Now, around the same time, we also started to do more longer-term experiments. I got sick and tired of people asking me, is it growth or is it aneurysmal dilatation? And I'm a surgeon, so we're dumb and we do things a dumb way. So I thought, if I put a graft in a lamb and I follow it until he dies and it doesn't form an aneurysm, they can't complain about that. So we're 8 years into this 10-year experiment, and we've discovered a lot of interesting things. One of the interesting things is that these neovessels really do not only look like native vessels, but they behave like native vessels. So if we look here, this is a native IVC, this is a neovessel. Both have a laminated structure. They're lined with endothelial cells surrounded by concentric layers of smooth muscle cells, but they're also vasoreactive. So here we're looking at how a neovessel compares to a native vessel when we expose it to vasodilators and vasoconstrictors, so that the curves overlap. So they're structurally and functionally quite similar to real blood vessels. Another thing we're very interested in looking at is the extracellular matrix and its effect on the biomechanical properties of the cells. Here we're looking at 4 different time points, a picoserious red stain under black light, which shows the scaffold nicely. And what we see is that the scaffold degrades by about 6 months, like it's supposed to, and we see lots of collagen. But initially the collagen's green because it's very mature. It turns orange as it starts to mature and turns red when it's really mature, and it becomes much more highly organized. And during this process, what happens is our scaffold goes from being very stiff relative to the vessel that it's implanted to highly compliant and very similar to the compliance of the vessel of which it's implanted. And this provides our grafts with what's called compliance match, which winds up being very important to performance. So this is this is an angiogram of the inferior vena cava. We give a bolus of IV fluid to increase the flow of blood, and what happens? That graft gets bigger, and that's exactly what happens with our own blood vessels. In fact, the graft could increase in size by almost 50% just by changing the flow of blood through it. And when you compare that to Gore-Tex, the standard of care, when you have compliance match and you increase the flow of blood, your blood vessel, your graft, just gets bigger and it can accommodate the flow of blood. If you do the same thing with a Gore-Tex graft, it can't get bigger, so you develop a relative stenosis. And if you look at this computationally, what you see is when you have a Gore-Tex graft implanted in a blood vessel and you increase the flow of blood, you develop this stenosis, which increases the pressure gradient, which increases the power loss. But look at what happens to the blood distally. You actually get reversal of flow. This reversal of flow increases transit time, and that causes blood to clot. And that's the most common complication that people typically experience after this operation. Now, if you have a compliance match graft, you can avoid all these problems. When you give that bolus of IV fluid, the graft simply gets bigger. You don't develop the stenosis. You don't have the pressure gradient. You don't get the power loss. Growth, I keep coming back to this. This is what I've been harping on for 20 years. Yes, lambs get bigger over time. Yes, they get fatter over time, but the grafts also increase in size. But it's tricky because remember, we're talking about these are compliant grafts. So if you just look at diameter or area, that's a difficult way to try and measure growth because you have to have exactly the same pressure in order to compare things over time. In fact, these 2 measurements are taken of the middle of the tissue-engineered vascular graft 5 years after implantation. This is before a bolus, and this is after a bolus. So looking at diameter area is not the way to look at growth because they're susceptible to compliance differences. Length, however, is compliance independent. And look what happens when we look at the length of these grafts over time. It increases in size and then plateaus, and it plateaus when the animals mature and stops growing. So if you want to look at growth capacity, you really need to look at length, not area. Another thing we noticed about these grafts is that they don't calcify, and that turns out to be very important. Ectopic calcification or dystrophic calcification affects virtually all biomaterials, and it's a particularly bad problem in congenital heart surgery. And here we see CAT scans where we can use a thresholding method to detect calcification, and we're looking at tissue-engineered vascular grafts versus Gore-Tex grafts, and the Gore-Tex grafts are calcified 5 years after implantation of lamb, while the tissue-engineered vascular grafts are not. In parallel, we've continued our mouse work and tried to understand mechanism, but now with a goal of rational design. How can we improve the performance of our vascular grafts, or more specifically, is there anything we can do to reduce that incidence of stenosis? And we discovered a whole variety of ways of reducing the incidence of stenosis in our animal models, but 2 were really ripe and ready for translation to the clinic. One thing we discovered is that cell seeding inhibits stenosis in a dose-dependent fashion. So simply by putting more cells on the scaffold or saturating the scaffold, we could reduce the incidence of stenosis. And the other one was interesting. There's a medicine called losartan. It's an angiotensin II receptor 1 blocker. It's normally used for blood pressure modification, but it also affects the release of monocytes from the spleen. So it turns out when you implant our scaffold, there's a foreign body reaction. The monocytes come from the circulation. They infiltrate the scaffold. They form macrophages. These macrophages are what drive the stenosis. But what we could do by administering angiotensin is we could actually block the release of the monocytes from the spleen, which reduce this. And by combining cell seeding with the use of losartan in our animal work, we could actually eliminate the development of stenosis. So we shared all this information with the FDA. And about 5 years ago, began our next FDA trial. It was designed to be an exploratory confirmatory trial. In the device world, that's like a phase 1, phase 3 study in the drug world. So in other words, you finish a study and you've got the data that you need to get approval if everything goes well. And we enrolled our first patient about 5 years ago. And we performed an interim analysis after the first 5 patients, which were implanted in a staggered fashion, and demonstrated that we had significantly reduced the formation of stenosis in our grafts, and then initiated the next phase of our trial, which is ongoing now. Now, of course, as soon as you show that you got rid of stenosis, the next patient has stenosis. So that's just the way life works. And in the interim, the FDA comes back and says, "Oh yeah, I know you are doing this clinical trial, but we're going to change the rules on you now." In the trial, we had said that we were going to compare our results of the tissue-engineered vascular graft to historical controls, and the FDA came back halfway through the trial and said, "Well, you really need to have a concomitant standard of care to add to your travel arm." So we added an arm of the study where we were looking at Gore-Tex, and the FDA actually gives you good advice, and it was exactly the right thing to do. And what we discovered is that when you look at performance critically, you find problems that you didn't know about. So if you read the literature about Gore-Tex, it's supposed to be smooth sailing. You're not supposed to have any problems when you implant a Gore-Tex graft for the first few years. But in fact, when you analyze it as carefully as we look at the tissue-engineered vascular graft, the performance was actually fairly similar. One of the things we discovered is that the Gore-Tex grafts actually lose about a third of their diameter over time, which is something that wasn't widely appreciated. We also continue to follow our TVG patients in our clinical trial, and we're able to nicely demonstrate growth using serial MRI. And then just this past year, papers started to come out in the literature showing how Gore-Tex actually has all kinds of problems when you implant it in the Fontan circulation. For 20 years, I've been talking to my colleagues in congenital heart surgery saying, wouldn't it be great to have a graft that grows? You wouldn't have to redo your surgeries when your patients outgrow the grafts. And they'd say, well, we really don't have to do that very often. What we actually do is we put in these oversized grafts and we let the kids grow into them, and it's a really good strategy. And the strategy worked for about 10 years, but then it didn't work anymore. So now there's this emerging body of literature that shows that virtually everybody outgrows their Gore-Tex grafts, and there's a rise now to perform redo surgeries on all these kids during the teenage years. So there actually is a need for a graft with growth capacity. We continue to look at calcification. These are images from patients more than 10 years out after the study, comparing Gore-Tex to TVG, and again, still very resistant to ectopic calcification. Growing body of clinical literature showing that virtually all the Gore-Tex materials calcify in this particular implantation. And then we've been looking at compliance. We've been using speckle tracking to look at our echo data, and you can see when the grafts initially implanted, they're not very compliant, but over time, the wall starts to become much more supple and changes the area of the graft as the pressure changes over the cardiac cycle. And this is turning into another clinical issue as physicians are starting to look at exercise. What they're seeing is that the lack of compliance results in the development of a transient pressure gradient, sort of what we showed you before in the LAM work. Now, ultimately, the Fontan operation is a palliative, not a curative operation. You don't create a new ventricle for these patients. You simply bypass the hypoplastic ventricle, which means these patients have very abnormal hemodynamics. They have venous hypertension and a low cardiac output. And over time, this causes end organ disease. And believe it or not, one of the most common causes for death in children that have Fontan operations isn't their heart failure, it's actually cirrhosis. The liver is very susceptible to this venous hypertension, and it causes cirrhosis, and these kids either go on to die or need liver transplants. And what just came out in this past year is that if you had your Fontan operation done with a Gore-Tex graft, you actually had a 7-fold increase in the chance of developing cirrhosis 15 years after surgery. So again, a big need for a better graft and an emerging field of this redo Fontans. So we have 2 more patients to enroll in this year, and then we are going to submit what's called the humanitarian device exemption. We have orphan designation for our tissue-engineered vascular grafts, and fingers crossed, hopefully we'll be able to give them data that merits approval. And hopefully through this talk, you get a sense of some of the pieces of the puzzle and bring the technologies from the bench to the bedside. I appreciate your attention, and I'd be happy to answer any questions.
Chris, great talk. Thank you. Shelley Payton Tufts. I'm wondering about the stenosis, the narrowing. Is that symptomatic in the patients, or is it just picked up on the angiogram, and that's how you found it, which is why the FDA sort of pulled the study?
They were all the cases of stenosis that we detected were picked up incidentally, so the patients weren't stenotic. But Fontan patients are very susceptible to stenosis, so you have to be super careful. So we do a ton of imaging and still will do angioplasty in certain situations if we're worried that the patient is going to be in danger from the stenosis. Yeah.
It's fascinating work. I guess I have a question about the larger kind of entrepreneurial side of this in terms of, you're talking about orphan designation, like the number of patients and market analysis and how that goes into your ability to do this. I mean, it's a long, long process, and I can see it as a first of many proof of concepts to get to a larger market, but how did that influence how you were able to push this forward on a commercialization front?
Sure. So a couple things to address. So I had an allergic reaction to working with industry in the beginning. When I was in the laboratory with Vicanti and Langer, we actually didn't have any NIH money at a Harvard lab, which is kind of funny. Everything was funded through, they had created a new matter patent. They had actually patented tissue engineering. They literally copied all the pages from the Human Atlas and used it as an appendix. And they said, we can make this. And that got serially sold for hundreds of millions of dollars. So our lab was basically the R&D for this company called Advanced Tissue Sciences. Mm-hmm. And on one hand, I thought it was really excellent. It was nice to have this great funding source. But the company made several decisions that I thought shouldn't have been done from a scientific reason, mm-hmm. and that never sat well with me. I kind of feel like tissue engineering would have been much further advanced if they had never been involved. And specifically, they didn't want to use autologous tissue, that they said, we can't figure out any way to create a commercial entity that makes tissue locally. We need to have some central facility where we make tissue and send it out. And I just thought that was the death knell for the company, and ultimately it was. So I avoided industry like the plague. I've done everything through federal funding and philanthropy. And so what's cool is now we're hopefully close to clinical approval. And now I do have to get involved with industry because you need to market this. Mm-hmm. Now, if you go to a company or you go to an investment firm, you say, hey, I've got this great idea, and it's going to help 1,000 pages a year, which is the number of Fontans that are done per year, they quickly gloss over and thank you for your time. But what's happened are a couple things just in the last year which has really changed that. So one of the things that occurred to me is, while I've done all my studies to look at this graft as a Fontan, I've actually looked at it in a variety of different circulations. We've looked at it in the pulmonary circulation, we've looked at it in the venous circulation, we've looked at it in the portal circulation, and there are no products available for use in those circulations. Mm-hmm. And so they're basically ignored. What people do is they take products that were designed for use in the arterial circulation, they use them in the venous circulation or the pulmonary circulation or the portal circulation, and then they wonder why they don't work so well. And so when you cobble all that together, it actually creates a rather sizable market. There's probably a market in excess of 50,000 patients per year in the US alone for this graft. So that was one way around it. The other thing that happened is when you're pricing things, if you're going to price a product, the way insurance companies look at pricing is they want to see how much money they're going to save because of the advantages of your product. Mm-hmm. So now, because everybody in the U.S. now is starting to redo Fontan surgeries, that means that the price of our graft should be the cost of a Fontan operation, or at least something approaching that. Fontan operations are a couple $100,000. So you can save significant amounts of money for the insurance company if you have a product that can grow and prevent the need for these applications. So long-winded way of saying that I stayed away from industry until the point that we need to make this technology available. And then just per chance, these other things sort of happened, and we identified bigger markets and attractive pricing schemes.
Thank you so much.
Joyce Wong, Boston University. Chris, that was really a beautiful talk, and thank you also for sharing that you really had these wonderful parallel careers of you and Shinoka and Humphrey coming together, I think, and it really epitomizes, I think, what Hub is trying to do. So my question to you is you had the comparison with the Gore-Tex, but I'm wondering if you've actually compared autograft, and it's probably not possible to do in pediatric, although I know they'd use pericardium, but I'm wondering in either Jay's model or something, do you also, if you have an autologous graft, as you know, like mammary arteries or things like that, do you also get the kind of stenosis, or maybe you don't get as strong as an inflammatory response? I'm wondering if you can just comment on the autograft aspect, you know, thinking about smaller diameter as you're pushing for them, just wondering your thoughts on that.
Yeah, so the beauty of the Fontan operation is you have a built-in autologous control, so you can, we actually do that with every patient. And there we go. So when a patient has this operation, it's actually a series of operations to create what's called the Fontan circulation. And in the Fontan circulation, you take all the venous drainage from the body, all the blood from the superior vena cava and the pulmonary artery, and connect it to the pulmonary artery. Superior vena cava, inferior vena cava, pulmonary artery. Now, the superior vena cava is positioned in such a way that you can do a primary anastomosis. So every one of our patients has a built-in autologous control, and they don't see the same degree of stenosis that we see. And they do increase in size over time. But it provides that control so we can say, oh, look, when this increases in size, it matches this, so we know we're growing and not forming an aneurysm.
So then to follow up, so then would Jay, in his model, would he predict that? What are the parameters he would do to then explain that where you don't see stenosis?
Oh, yeah. So what does the modeling tell us causes stenosis? So neotissue formation is inflammation-driven and mechanomediated. So initially it's the excess inflammation that's driving it's excess inflammation that's driving stenosis. So reducing the degree of inflammation can reduce the incidence of stenosis.
So the assumption is that if it's autologous, you just would have reduced inflammation because it's autologous? Is that
yes.
right? Okay.
Yes, and same reason for the calcification.
Okay, thanks.
All right, good morning. Lola Eniola, UIC. Thank you so much for that talk. I have many questions. I'll narrow it to two. Excessive inflammation causes the stenosis, but inflammation is needed for remodeling. I understand that you're using the angiotensin to sort of modulate that. How do you get the sweet spot?
Yeah. Well, the dosing that we use just happens to hit that sweet spot. But I don't think, I think one of the things that I'm starting to appreciate now is we're heading into the first real examples of precision and personalized medicine.
Yeah.
And I think that there's going to be a portion of patients that are going to have more stenosis.
Yeah.
There's going to be a portion of patients that have less stenosis. We all have our own immune systems. We've
Yep.
all been exposed to different things. We all have different genetics. And so one of the things that we've also been working on are developing molecular imaging techniques to try and quantify the inflammation to see if we can be a little more targeted in who we treat with what. to modulate the inflammation. So, for example, if you use FDG PET scanning, FDG PET scanning looks at glucose metabolism and the inflammatory cells that infiltrate the scaffold, the macrophages, are highly metabolically active. If you do an FDG PET scan of one of our animals after implantation, it lights up like a Christmas tree. And so we're looking to see if we can quantify things precisely enough that we can use more precision medicine.
Very cool. So I
The,
see.
sorry, the way we got to that conclusion was that the mouse work actually turned out to be really helpful. If I implant a tissue-engineered vascular graft into a monocyte-specific conditional knockout where I can literally get rid of all the monocytes, you have a scaffold and no inflammation and no neotissue formation. So you have a biodegradable scaffold that'll go on to rupture and bleed. But if you get excessive inflammation, you get the stenosis, and if you hit that Goldilocks spot, you get the nice neovascular.
Yeah, no, I think it's cool if you can track the glucose to the sweet spot. You have this long runway that shows you started this work in 2000 and you're projecting 2030.
Mm-hmm.
That's a long runway.
1995, even worse.
Can you give us a sense of what you know now that if you had known in the beginning would have accelerated you to this point?
Yeah, no, great question. So we were the first ever to get FDA approval. So we're the first to implant a tissue-engineered vascular graft in the human and first to get FDA approval. And so that's a learning process. Not only were we learning, but the FDA was learning. And the underlying assumption had always been, once you get through once, the ability to cut and paste will really accelerate the process. And I truly believe that will happen. I think one of the things that I'm really excited about is computational modeling and the notion of CAD-CAM. So computer automated design, computer automated manufacturing. Jay and I are working on a grant right now. We're just about done executing a grant where we do what's called canvassing the search space. You use set-based design to try and get a sense of what you can manufacture, using your methodology and what the results are. And then we do our optimization computationally. So rather than doing an infinite number of experiments where you look at every combination and permutation, you do a handful of experiments, let the computer do the heavy lifting, and then validate it. And that's coming along quite well. And I think that will really accelerate development.
Thank you so much. This is a great time to stop and thank. Oh, I'm sorry.
Maybe it's a quick question.
Yeah.
And I was like, why is Karen looking at me?
Sorry to hold you from your break, but thank you for an amazing talk. I'm Anna Gaffney. I'm a PhD student at University of Chicago, and I'm a bit new to, I've just started kind of working in a vascular surgery lab, and I find the research really fascinating. I'm interested to know if you see this kind of implementation being used on adult patients, or if there's any size dependency on whether this would work. And also if you see this type of graft and tissue engineering being able to be used on things like stents or something that can be put inside of already their channels and organs.
Yeah, thank you for your question, and great choice. You'll have a wonderful career doing cardiovascular tissue engineering. It's a lot of fun. Yes, absolutely. There's a number of people working in the adult space too, and probably the most exciting is Laura Nicholson's work, which just got FDA approval this year. She sort of took the opposite approach. Laura started a company, had huge amounts of investments and did great work. They got FDA approval, but interestingly, they got FDA approval for a tiny market when all was said and done. They got approval to use their grafts in trauma patients who have no autologous tissue that need a emergent graft. And that's very exciting. Having an FDA-approved product is great for the field, and I have extraordinary admiration for work. And I think absolutely that there's lots of great work going on now, in addition in the vascular graft space, but also in the heart valve space and even the whole heart space. So.
Thank you.